All right, welcome everybody to Chem 313, lecture number 11. To review, we covered quite a bit of ground last time. We were talking about membranes and specifically membrane proteins. And membrane transport. And we were largely building up to today's material with all of these lectures. So you'll recall that last time we said that membranes, last two lectures actually, these are bilayers of amphipathic lipids. So the way those are often drawn is like this, where we've got the polar head, and we've got the nonpolar fatty acid tails, or in the case of sphingolipids, they're hydrocarbons that look like fatty acids but aren't exactly fatty acids. And as long as they are amphipathic and they're rectangular in cross-section, that implies that they will form bilayers when you disperse them into water due to the hydrophobic effect. So in addition to membranes themselves and their structure, we also discussed another couple of topics. One of those was fluidity, which is the liquidness, if you will, of a bilayer. And we said that this varies with temperature. And so as you might expect, just as solids turn to liquids if you heat them up, as temperature increases, fluidity also increases. And we said that cholesterol has an important role in moderating fluidity. And fluidity is very important because it feeds into the function of membrane proteins, such as channels and transporters, which are proteins embedded in the lipid bilayer and must undergo conformational change. So that led us into the topic of membrane transport, which refers to solutes crossing lipid bilayers. They don't necessarily pass right through a lipid bilayer, but membrane transport is the process, the class of processes by which solutes cross lipid bilayers. And we were able to distinguish three different classes of membrane transport. We have active transport, which is transport in the direction in which the free energy of moving the solute from side A to side B is positive, so unfavorable. And active transport, by definition, requires work. And that can either be supplied by ATP or by transit of something else with its gradient. So in other words, along the direction in which its delta G is negative. And it usually involves transporter proteins. Or channels. We also talked about facilitated transport, which is transport in the direction of the free energy from side A to side B being negative, so favorable. So this is transport that will occur spontaneously by itself, but which is catalyzed, and that requires a channel. And you can think of a channel as being sort of like an enzyme, but for transport through a lipid bilayer. The last class is passive transport, and that's transport for which the free energy of transit is negative, so it occurs spontaneously, and the molecule just goes directly through the bilayer. And that's for the most part limited to small and nonpolar compounds. All right, so how do you know which direction has a free energy of transit that is negative? And the answer is you have to use the Nernst equation, which is that the free energy of transporting a solute from side A to side B is going to be equal to RT natural log concentration of whatever solute you have on side B divided by the concentration of that solute on side A plus Z times F times psi B minus psi A. Z is the charge on the solute X, including the sign. F is a constant, 96,500 joules per volt per mole. And these are the voltages on sides B and A, which are typically going to be provided to you in a problem like this. So the Nernst equation looks intimidating, but it's actually quite simple, right? If you're talking about a solute that has no charge, so it's not an ion, delta G of transit from A to B will be negative if the concentration on side B is less than the concentration on side A. So in other words, it's spontaneous to go from high to low concentration. Exactly how spontaneous, you have to do the math. If the solute is an ion, then you have to do the full calculation to find out what the delta G is. Now, the value of the delta G doesn't have anything to do necessarily with the type of transit that you have. I mean, if the delta G is positive, you would need to have active transport, okay? But the value of delta G doesn't have anything to do with whether you need catalysis to cross the membrane. So even if delta G is negative, if you have an ion, you need a catalyst, you need a channel to cross the lipid bilayer because ions simply will not cross a lipid bilayer. So active and facilitative transport, regardless of delta G, both of these involve channels or transporters. And another point that I want to make, a final point about active transport, is we say that active transport is transport in the direction of delta G being positive. But that doesn't mean that active transport has positive delta G. That would mean it's impossible to do without work. The whole idea of active transport is you couple transport to something with a negative delta G so that the net free energy is negative. So this lecture is going to talk about cell signaling. And we reserve this topic for right after we talk about membranes because I think it gives a pretty good illustration of why membrane biology and structure and function are important. So this is in the textbook, chapter 13. If you're using the Open Learning Initiative text, there is no module in the Open Learning Initiative text dealing with cell signaling. So unfortunately, you're either going to have to partner up with somebody who's got the textbook, or you can probably find several reasonably good online resources with a little bit of snooping around. But here's the good news. You're only expected to know a few things. The main reason we talk about this subject is it sets us up for the metabolism unit of the course. For practice, you can find practice problems related to cell signaling in homework 5 and in worksheet 7. Okay, cell signaling, what is it? Cell signaling is essentially how do cells respond to changes in their environment. In order to secure life. So, for example, there is a cell that's growing on some sort of a food source. So we've got bacteria that are growing on some kind of food source, and we're saying that there's a change in the environment. So the food source, we'll say, suddenly goes away. The organism that it was growing on died, or there is some sort of a chemical change in the environment. Something happens so that a food source suddenly disappeared. So what's going to happen? Well, what needs to happen is that if the cells did nothing, they would quickly die because what they have been eating is going to disappear. So what ends up is happening is that cells have to make changes, right? So for example, if a food source goes away, cells may stop taking in energy from the surroundings and may conserve whatever energy they have. All right, that's just a loose example. So the whole process of going from the change in the surroundings to the change in physiological response, we'll say change in cellular activities, which could be anything. That could be changes in biochemistry, changes in metabolism. It could be changes in behavior if you're talking about an animal. This whole process is cell signaling. And now is when we really get into biology instead of just structures of molecules in this course. So almost all cell signaling, most organisms undergo cell signaling to some extent, but it's most complicated in multicellular organisms like animals. And there are some entire organs in animals whose job is just to tell other cells how to react to changes in the environment, right? That would be the role of the endocrine system. So how this whole process works varies quite a bit from organism to organism and from environment or change to change, or we'll say from situation to situation. But there are some common features. So I'll highlight here what you should know are the common features. Okay, so you should know the common features of cell signaling. You should know how to identify them. Okay, if you're given a description or a diagram. You should be able to predict likely outcomes of signaling. Okay, what would a cell likely do in this situation? And then a general class of question that I'm fond of asking is to be able to predict how signaling would be different if certain signaling compounds, proteins, enzymes, etc., were different. So those are the things that I'm going to be testing you on for the cell signaling module, all of this stuff down here. You do not need to know the details of each pathway. So I'm not going to expect you to rattle off a list of everything that happens during insulin signaling. I think that's pointless. What you should be able to do is identify common features between insulin signaling and, say, glucagon signaling, be able to identify which feature of cell signaling is being shown in a diagram, predict what the outcomes of that signaling would be, which is going to depend on what the change in the situation is, and then predict how that signaling might be different if you were to change the function of one of the molecules that's involved in that signaling. All right, so let's take a look at all the different steps that are common to all signaling systems. All right, so there are some common components to all cell signaling processes. All right, what are they? The first thing is we need a signal. So what's a signal? So I'm going to define a signal as some sort of sign of a change in the situation or environment. Okay. In some cases, the change in the environment itself may be the signal, but not necessarily. So, for example, a hormone may be secreted. So hormones are simply molecules that are produced by some cells to send a message to other cells. So hormones are probably the most common form of a signal used in cell signaling, but that's not always the case. Okay, another example of a signal would be, especially in the nervous system, a change in the membrane potential across a lipid bilayer. That can serve as a signal as well. How does that take place? How does the change in potential across the membrane occur? By change in concentration of certain ions on one side. In some cases, something as simple as a change in temperature can function as a signal. So some of the heat and cold sensing mechanisms in nerve and muscle cells all boils down to a change in temperature. So that's an example of something where the change in the environment itself is the signal. In this class, we're going to mainly focus on hormones as our signals because most of the well-characterized cell signaling processes are based on hormone signaling. But you guys should be aware that cell signaling doesn't necessarily involve a hormone. Second component of cell signaling is we need some sort of a receptor. So what is a receptor? A receptor is a protein that senses the signal. Usually by binding, usually accompanied by some sort of a change in the conformation of the protein. In some cases, such as when there's no hormone involved, like when there's just a change in membrane potential, you get conformational change without binding. But since we're going to focus on hormone signaling, binding is pretty much a requirement for receptors to work. So receptors are usually, but not always, embedded in the outer membrane of the cell, the plasma membrane. And so they're usually transmembrane proteins. All the examples that we'll see are of transmembrane proteins as receptors. So an example would be the insulin receptor. We'll see that at the end of the lecture. Third component of signaling, this is the one that I think people have the most trouble with, is the so-called relay. So what's a relay? So a relay is a process for carrying a signal from the outside of the cell to the inside. And so how does this work? Well, usually it involves binding to or reactions with other proteins. So at this point, the signal is moving away from the receptor and the message is being carried by other proteins. And oftentimes, this involves release of molecules that we call second messengers. And what are second messengers? Second messengers are molecules inside the cell that diffuse through the cell interior and bind to targets of the signal. In some cases, second messengers are small molecules. We'll see an example of that, of a small polar freely soluble molecule. In other cases, second messengers are proteins. The fourth thing that happens in every cell signaling process is essentially the whole point of signaling, and that is what we would call target activation. And so this is activation, or in some cases, inhibition of proteins that carry out whatever cellular function it is that needs to change. Okay, so for example, conformational change in muscle proteins, myosin and so forth, that leads to contraction. All right, so what we've done is we've gone from a change in our environment, which produces a signal, to a receptor, to a relay, to activation of our target protein, and this produces the results that the cell needs in order to respond to the change in its environment. But there has to be a fifth step, and the fifth step is termination. Cell signaling is essentially always understood to be temporary. And so what happens in termination is we restore the previous situation and how do we do that? Well, usually there's more than one mechanism of termination, but typically it involves a return of all signaling proteins or molecules to their so-called default state. In other words, before the signal ever propagated. So those are our components of cell signaling. And on the next slide, we're going to look at an example of cell signaling. And the example that we're going to look at is epinephrine signaling. All right, so let's take a look at cell signaling using as an example epinephrine, which is also known as adrenaline. So you may know what adrenaline is. So this is a hormone that is released by the adrenal glands in response to a sudden stress. And the goal is to prepare an animal for sudden evasive action. This is something that's released in emergencies, and so in response to adrenaline, you're going to need to do things such as run. So think about it. What do you need if you need to run? Well, if you need to suddenly run, we need to have certain molecules available. So for example, oxygen. So what we will need to do is to have a good steady blood supply of oxygen, because muscle cells are going to need oxygen and the heart is going to need oxygen if you're going to have to flee from some sort of a danger, okay? But in addition to that, oxygen isn't enough. You also need fuel, such as glucose. And epinephrine signaling causes release of glucose from tissues that produce or store glucose, such as the liver. And so the release of glucose is what we are going to focus on when we look at epinephrine signaling. So let's take a look at what happens during the process of epinephrine signaling, and I'm going to point out every single step of the process or different common features of the signal as we go. All right, so for those of you who are interested in epinephrine, it's a molecule that's made from an amino acid. It's a tyrosine derivative. I believe that's what the structure is. And this is produced by the adrenal glands. And then it's secreted into the blood. So it circulates in the bloodstream and gets to cells such as muscle cells and liver, which are sensitive to epinephrine signaling. And so this molecule is going to function as our signal. All right, so what happens next? So what happens next is after epinephrine floats around in the bloodstream, it makes its way to its specific cell surface receptor. So this would be our receptor. The epinephrine receptor is a molecule called the beta-2 adrenergic receptor, beta-2AR. So the beta-2AR is an example of a type of molecule called a GPCR, G protein coupled receptor. So a G protein coupled receptor has 7 transmembrane alpha helices, which are not clearly shown in this diagram, but I'll show you a better structure on the next slide. And there's different types of G proteins, but the beta-2 adrenergic receptor is bound to 3 proteins on the inside. And these are called the G proteins. So the receptor is one molecule, and the 3 G proteins are 3 separate molecules. And so we've got alpha, beta, and gamma G proteins. The important one for our purposes is the alpha protein, which we're going to call that G alpha. And there's different types of G alpha proteins, but the one that's involved in epinephrine signaling is called G alpha S. All right, so it's the beta-2 adrenergic receptor itself that is the receptor. So our receptor is the molecule that directly binds to the epinephrine. The epinephrine binds in a little pocket on the outside of the cell. This is the outside of the cell at the top. And then what happens after receptor binding? So after the molecule of epinephrine binds to the receptor, the relay begins. So what happens with our relay? So what ends up happening is that binding of epinephrine on the outside of the cell causes some sort of conformational change, which is still not very well understood, in the shape of the receptor, so that the G proteins, which are stuck to the inside of the cell, change their conformation as well. So our relay involves a conformational change in the G proteins. And the reason these things are called G proteins is that they bind to GDP and GTP. So GDP, GTP, that's the same thing as ATP and ADP, except using a G, nucleotide instead of an A. So what ends up happening is that the GDP gets swapped out in favor of GTP on the G alpha S. And when that happens, the G alpha S protein, now that it's bound to GTP, moves away from the complex. So the G alpha S, we'll use a colon to indicate a complex, GTP complex, is going to then break loose from the beta-2 adrenergic receptor complex. And at that point, it simply scoots along the inside of the plasma membrane of the cell. All right, we're still in the relay phase of this reaction, by the way. So what happens next? So the G alpha S protein, after it breaks away from the GPCR complex, it binds to this other enzyme which is embedded in the membrane. Its name is adenylyl cyclase, or some textbooks called it adenylate cyclase, which is not strictly correct. Adenylyl cyclase is the official name, but you can just call it AC and everything is just fine. So adenylate cyclase is an enzyme, and what it does is it catalyzes this reaction. ATP forms a molecule called cyclic AMP plus PPI. PPI is just pyrophosphate, which is 2 phosphate ions stuck together. All right, so only when bound to G alpha S can adenylate cyclase carry out this reaction. So in other words, the G alpha S functions as an allosteric activator of adenylate cyclase. And it's such a potent allosteric activator that adenylate cyclase is essentially dead unless it's bound to G alpha S. Now we've released this molecule called cyclic AMP. Cyclic AMP is our second messenger. So cyclic AMP is a fairly small molecule that can freely diffuse inside the cytoplasm. And what it does is it gets released into the cell and it binds to other proteins and changes their function. So cyclic AMP is a second messenger, but its actual role is that it's also an allosteric regulator of proteins. And the most important protein that cyclic AMP regulates is a different enzyme that is known as PKA, protein kinase A. So at this point, we have made it all the way to our activation of our targets. So protein kinase A is our primary target in epinephrine signaling. Now, protein kinase A is an enzyme. We're going to talk about it in more depth on the next slide. But what protein kinase A does is it then turns on and off other cellular proteins. So phosphorylation of cellular proteins by PKA causes the cellular response to epinephrine. We'll talk about that more in a moment here. So that's our target activation stage. And so after that occurs, the cell is going to change its function in such a way that enables the organism to survive the acute danger. And then eventually the danger goes away, and then what happens? What happens is we have to terminate, right? So our termination is provided by this enzyme called cyclic nucleotide phosphodiesterase. So cyclic nucleotide phosphodiesterase destroys cyclic AMP. It catalyzes the conversion of cyclic AMP to regular AMP, which is something that's already normally floating around in the cell in millimolar concentrations. And then eventually AMP can be converted to ATP to restart this whole process. So this is the entirety of the epinephrine signaling pathway. Let's take a look at some of the steps of this process in a bit more detail. This is showing us a rough diagram of the beta-2 adrenergic receptor. So the actual structure of the protein looks like this. The N-terminus of the protein is on the outside of the cell. The C-terminus is on the inside. And the G proteins, and there's 3 of them, the G proteins all bind down here at the bottom of the receptor. So our epinephrine binds in this pocket at the top, and then the G proteins interact down at the bottom. So this figure down here at the bottom right is showing sort of a zoomed out view of what this molecule would look like. It's rotated slightly. But we've got our 3 G proteins here, alpha, beta, gamma. It's the alpha protein that does most of the work. So once again, binding of epinephrine to the beta-2 adrenergic receptor leads to G-alpha S binding GTP. And then after G-alpha S binds GTP, it breaks loose from the complex and moves along the interior of the plasma membrane until it binds to adenylate cyclase. All right, so I don't have a nice ribbon diagram of adenylyl cyclase to show you. I think the structure of the entire protein is not yet known. But this is the reaction that it catalyzes. ATP gets converted into this cyclic molecule called cyclic AMP, and this requires allosteric activation by G-alpha S. All right. Now, what does cyclic AMP do? So by itself, it's not a terribly useful molecule, but we said on the last slide that cyclic AMP is itself an allosteric regulator of other proteins, and it's also a second messenger. So I should emphasize now that not every second messenger is an allosteric regulator. Some of them are enzymes. But in this case, our second messenger functions as an allosteric regulator of other proteins. The most important protein that it binds to and activates is this protein called protein kinase A. So we need to define what a protein kinase is. A protein kinase is an enzyme that catalyzes a reaction like the following. A protein, some protein, a target protein, plus ATP react to give us ADP plus our protein again, but with a phosphate group attached to it. And so this process is known as phosphorylation. So what protein kinases do is a protein kinase attaches a phosphate to a protein, and normally where the phosphate gets attached is to either threonine, serine, or tyrosine side chains. On rare occasions, histidine sometimes will be phosphorylated as well. So what happens when you phosphorylate a protein is you go from, if this is the backbone of the protein, and we'll say that we're doing a serine, ATP functions as a phosphate donor, so our byproduct is ADP, and our protein ends up chemically modified so that if you look at the serine side chain again, now it's got this big phosphate on it. So in this case, we have what's known as a phosphoprotein. So phosphorylation of proteins is an extremely common means of regulating the function of a protein. Right? Now ask yourself, why would putting a phosphate group on a serine side chain or a threonine side chain, why would that lead to a change in the function of the protein? In fact, it's very, very common. Most organisms that aren't bacteria regulate protein function in this manner by attaching phosphates to the side chains of certain amino acids. So ask yourself that. Why do you think putting a phosphate on a serine, threonine, or tyrosine side chain might change the function of a protein, and how? All right? I'm not going to give you the answer, but I think you can figure it out if you think about it a little bit. So protein kinase A attaches phosphates to various proteins, ending up with proteins with modified function. So attaching phosphate like this, what it actually does to the protein, it completely depends on the protein. Some proteins are activated by phosphorylation, some are inactivated by phosphorylation. Some don't have any change in function with phosphorylation, but it causes other proteins to bind to them and do other stuff. So it depends on the situation what phosphorylation actually does. Now, a lot of the proteins that are activated by protein kinase A are enzymes. In fact, some of them are actually protein kinases. And these enzymes do things such as break down glycogen. So what's glycogen? We'll get to that in the next lecture, but glycogen is essentially glucose packed into a very small space inside the cell. And so one of the byproducts of protein kinase A activity is that glucose levels in the cell will increase. And that's important in muscle cells because glucose is going to be your energy source to do things like run. Now, let's say that the danger has passed, and what we have to do now is to terminate our signal. So our signal termination comes from cyclic AMP unbinding from our proteins, and any loose cyclic AMP floating around inside the cell is eventually going to make its way to this enzyme called cyclic AMP phosphodiesterase, which destroys cyclic AMP and gives us AMP, regular. That's our termination step. Now there's a second mechanism of termination that I didn't show in the slides. And that is, remember, adenylyl cyclase, which starts this whole thing by producing cyclic AMP, it's activated by G-alpha S when bound to GTP. Well, it turns out that the G-alpha S is an enzyme. It's just an incredibly weak, slow enzyme. So G-alpha S will slowly hydrolyze GTP to give us GDP. And when it's in the G-alpha S GDP form, it can no longer bind to adenylate cyclase, and it returns to the beta-2 adrenergic receptor, and the whole process restarts. All right. An important aspect of cell signaling that you see in most but not all signaling systems is amplification. So what's amplification? Let's take a look at the process that happens throughout this entire cell signaling pathway. Let's say that we have one molecule of epinephrine binding to an epinephrine receptor at the surface of our cell. All right, good. So one epinephrine molecule binds to one beta-2 adrenergic receptor, and that causes the release of one G-alpha S, and the release of the one G-alpha S causes activation of one adenylyl cyclase. Okay, so far so good. However, notice this. Adenylyl cyclase is an enzyme, and enzymes can turn over or catalyze reactions multiple times while they're activated. So that means from your one G-alpha S, you'll end up getting maybe 20 molecules of cyclic AMP. All right, so your cyclic AMP is going to bind to protein kinase A, and I didn't tell you this, but it takes 2 cyclic AMP to activate protein kinase A. So that'll give us 10 pKa molecules. All right, so we lost a little bit of signal there. But nonetheless, protein kinase A will activate this other enzyme, phosphorylase kinase. Each of those 10 can activate roughly, on average, 10 molecules of phosphorylase kinase. So now from our one molecule of epinephrine, we have 100 active phosphorylase kinase molecules. And then in turn, each of those can activate 10 molecules of glycogen phosphorylase, so that our one molecule of epinephrine gives us 1,000 active glycogen phosphorylases. And then each of those 1,000 glycogen phosphorylases can release roughly 10 glucose molecules from glycogen. So from one epinephrine, we end up releasing roughly 10,000 molecules of glucose. And this is entirely due to the fact that enzymes can remain active for a period of time, and they can turn over multiple times before they become inactive. It's not just a one-to-one binding. As long as you have activation of an enzyme, your reaction can occur many times. All right, so what I've done here is ask you guys a couple of practice questions. These are sort of, questions that are somewhat like what I would ask on a quiz or a test. So I would recommend that you pause the video and try to answer these questions on your own before I give you an answer here. All right, so for the first one, how would epinephrine signaling be different in a cell in which cyclic AMP-dependent protein kinase, or pKa, had a mutation that made the KD for cyclic AMP binding unusually high? All right, so what we're saying is that pKa binds cyclic AMP weakly. So what do you think? How would the signaling be different if pKa bound to cyclic AMP weakly? Well, remember, the KD is the concentration of ligand that gives 1/2 of your protein molecules bound. And so if the KD goes up, because that's what weaker binding means, that means that you would need a higher concentration of cyclic AMP to get pKa active. Okay, we need a higher concentration of cyclic AMP, but where does cyclic AMP come from? It comes from adenylate cyclase. So that means adenylate cyclase will have to work longer before cyclic AMP levels get high enough. And so to answer the question, how would signaling be different, you would have a delayed signal. It would take a bit longer to get up to the critical level of cyclic AMP, and overall signaling would probably be weaker. You would probably have a weaker response, because if it takes longer for AC to generate enough cyclic AMP, there's a chance that some of our G-alpha S molecules may have hydrolyzed GTP and gone back to the receptor before enough cyclic AMP had been generated. So we would have a weaker response. Secondly, there are two mechanisms of signal termination in epinephrine signaling. What are they, and why do you think two mechanisms exist? Well, we already talked about that on the last slide. The main one is the cyclic AMP phosphodiesterase. And what that does is it destroys cyclic AMP, so it causes your second messenger concentration to decrease. And the other one that we said is hydrolysis of GTP by G-alpha S. And once it hydrolyzes GTP, it returns to the beta-2 adrenergic receptor. And so those are our two main mechanisms of signal termination. But the second part of this question is, why do you think two mechanisms exist? Why two? The answer is quite simple. It's always good to have redundancy in case there is some sort of error in one of the signal termination mechanisms. So say, for example, that you got an epinephrine signaling, but you had just eaten some poisonous herb that inhibited cyclic AMP phosphodiesterase. Well, in that case, you'd be in trouble because your epinephrine signal would never stop, and you would be on high alert indefinitely. And high levels of epinephrine for prolonged periods of time are very bad. It's rough on your heart, it's rough on your kidneys, and this is something that would definitely be unfortunate. So in this case, we would still be able to terminate the signal just by the hydrolysis of GTP by G-alpha S. So this is sort of a safety feature. And evolution has built safety into most organisms. Thirdly, which step of signaling would be affected if the beta-2 adrenergic receptor had a mutation that greatly increased its affinity for G-alpha protein binding? Which step of signaling? I'm going to tend to say the relay. because that is the point at which the signal gets carried into the cell, and it's the interior of the cell part of the signaling that's carried by the G alpha protein. And so what would happen is that if the binding of G alpha protein to the beta 2 adrenergic receptor was stronger, this would also weaken our signal by making the relay less likely to happen. Why less likely to happen? Because if the G alpha protein binds to the receptor very strongly, it's less likely to break loose even once it binds to GTP. All right, one more example. Insulin signaling. So insulin is a hormone that is released by the pancreas of animals when glucose levels in the blood get too high. And not even necessarily dangerously high, just higher than they would be fasting. So the goals of insulin signaling, all right, these are the results that we expect to get. Any cell that uses glucose, we want to take in glucose from the blood, otherwise it's going to end up going out through the kidneys, and store glucose in the form of such molecules as glycogen or fatty acids. And we're not going to talk about how glucose is converted to fatty acids in this course, but just understand that it's possible for that to happen. So that's what we want. The whole process of insulin signaling is the process of going from here to here. We start with an insulin signal when blood glucose levels get high, and we end up taking in glucose and storing it. So let's take a look at how that works. All right, so our signal is insulin. So insulin is a hormone. It's actually a small protein. And what insulin is going to do is it's going to bind to its receptor, the insulin receptor. All right, there we go. So the insulin binds to the insulin receptor. The insulin receptor is a different type of receptor than the beta 2 adrenergic receptor. So the insulin receptor is a type of receptor that's called a receptor tyrosine kinase. It's both a hormone receptor and a protein kinase. So most receptor tyrosine kinases are inactive until the hormone binds. And then you can think of the hormone acting as an allosteric activator. So what happens when the insulin receptor binds is the receptor tyrosine kinase changes to an active conformation. And so what does a protein kinase do when it's active? Well, it attaches phosphate to proteins. And for receptor tyrosine kinases, what they first do is they attach phosphates to themselves. So the enzyme actually puts phosphate on itself, and what that does is it changes the Km, specifically it decreases the Km for other proteins. And so after the receptor has phosphorylated itself, these other target proteins start to bind. So these are the other proteins. All right, so these become phosphorylated. And now our signal has gone from the outside of the cell to the inside. So this would constitute our relay. And most of these other proteins that are phosphorylated, we're not going to talk about them in any detail, but most of them are either enzymes or they function as allosteric regulators of enzymes. So there's a whole chain of events that I'm going to kind of skip over right here. But let's move on to the results of signaling, right? So our activation of targets, we can kind of list a couple of events that occur. So the first target protein that gets activated is this glucose transporter called GLUT4, glucose transporter 4. So what is this? It's a transporter protein that normally sits on these little vesicles that are stored inside the cell. But in response to insulin signaling, these get moved to the plasma membrane. So that now in our plasma membrane, we have a whole bunch of these GLUT4 channels ready to take glucose in from the outside of the cell. So that checks our box for glucose being taken into the cell. So we've already accomplished one of our goals. So another goal, which is not shown in this diagram, but I'm just going to write it out to tell you about, is that there is an enzyme called glycogen synthase, which synthesizes glycogen. And it becomes activated. All right, it becomes activated in response to an insulin signal by being phosphorylated by protein kinases, and also because there's an inhibitor protein that gets phosphorylated by protein kinases and gets inhibited. We're not going to go into the details of that. So by glycogen synthase being activated, that means that glycogen levels will go up. And that's a form of storing glucose. And then things can get very complicated if you want to go down the list. So in response to some of the proteins that the insulin receptor phosphorylates, there are changes in gene expression, right? So we said before that one of the other things that glucose or that insulin signaling tends to promote is fatty acid synthesis. And that's a result of more long-term changes in what kinds of enzymes are present in the cell, and also which kinds of enzymes are activated. And so that's our other sort of longer-term outcome, is that we get changes in gene expression, and in some cases even cell differentiation. All right, lastly, we need to have some mechanism of termination. So our mechanism of termination involves a phosphatase. And so that's this enzyme here, protein tyrosine phosphatase 1 beta, PTP1 beta. So a phosphatase essentially reverses the reaction of protein kinases. So a phosphatase, we said protein kinases are enzymes that put phosphates onto proteins. Phosphatases take the phosphates off proteins. Including the insulin receptor itself. And so if we remove all the phosphates from the insulin receptor and all the other proteins that it phosphorylated, we kill our signal. Okay, so we're not going to talk much more about cell signaling. I think it's a nice little self-contained chapter, but where we're going with this in the future is our unit on metabolism. And cell signaling is very important for regulating metabolism. So that's when the cell signaling unit is going to become more important and more relevant, but we're not quite ready to go there yet. So our next topic, before we can talk about metabolism, is we need to introduce a new class of biomolecules, and that's going to be the carbohydrates. Luckily, carbohydrates are a very easy subject. So that's our next topic, and I will see you guys in lecture 12.